Okay, so what I want to do is pick up where we left off, and we left off with nucleic acids. So I gave you just a really, really brief treatment of nucleic acids right before the exam, and it was super rushed. And nucleic acids are important because they serve a singular function, and there are no other molecules in our body that serve the same function as the nucleic acids do. So the function of the nucleic acids is information storage. When we want to pass information to succeeding generations, we do that with nucleic acids. Now, the type of nucleic acid used between organisms can vary a little bit. The vast majority of the biological world, however, uses DNA. There are some things like viruses that use RNAs, their genetic information, and something called a retrovirus, like the AIDS virus or HIV. But most everything else uses DNA. And what's cool is the nucleic acids are conserved between all organisms. That is, the structure of our nucleic acids is the same for bacteria, for protists, for plants, for animals, for fungi. All of us use the same basic molecular shape. So, like the other biologically important molecules that we've been talking about, this is a polymer. It's a molecule that consists of repeating subunits. And the repeating subunits for this molecule are called nucleotides. And each nucleotide consists of three basic things. The first of which is a 5-carbon sugar. And that 5-carbon sugar is going to vary between nucleic acids. DNA uses a 5-carbon sugar called deoxyribose, and RNA uses a 5-carbon sugar called ribose. Hence, DNA, deoxyribose, or deoxyribonucleic acid, or RNA, ribonucleic acid. But all of them have this 5-carbon sugar. It kind of provides an attachment point for the other two really important groups. So the next part is a large phosphate group. Now, phosphate is important because it's a high energy molecule and it carries a charge. And we're going to take advantage of this structure when we create another molecule called ATP. And ATP is just chemical energy. It's a way for us to store a little bit of chemical energy, take it someplace, and then do some cellular work with it. And then the last thing is something called a nitrogenous base. And this is where the meat of the work is done by our nucleic acids. This is what actually stores information. The nitrogenous base exists as two different types. It can be a 6-carbon ring, or it can be another type. And this would be like an example of the T. So there are 4 nitrogenous bases. There's A's, T's, G's, and C's. 2 primary classes of nitrogenous bases, either purines or pyrimidines. And the purines all have the same structure. They are, instead of just a 6-carbon ring, they are a 5-carbon ring. Well, it's kind of carbon. It's really carbon and nitrogen attached to a 6-member ring, like this. So this is what our purines look like. And purines are either going to be A or they're going to be G. You can also have something called a pyrimidine. And pyrimidines are just a 6-member ring, like that. And these are either going to be T or C. Or in the case of RNA, they don't have C, or excuse me, they don't have T, so instead they have uracil, which is U. But this is what a nucleotide looks like. It's a sugar attached to a phosphate attached to a nitrogenous base. So this is our sugar, nitrogenous base, and this is our phosphate. Now what happens is we line nucleotides up in a line, and we covalently bond a phosphate to a sugar. So the backbone of our nucleic acids consists of repeating phosphates and sugars. And that bond is called a phosphodiester bond. So a nucleic acid is just repeating subunits of nucleotides that are all connected with covalent bonds. Now, the question is, how is it that information is stored in nucleic acid? And the answer is, it's stored as the sequence of nucleotides. Now, we can divide up the long DNA molecule into discrete regions called genes. And a gene, G-E-N-E, codes for some specific protein. And that gene region will have a unique, non-random sequence of nitrogenous bases. And those nitrogenous bases code for amino acids. And then again, the link between phosphates and sugars is called a phosphodiester linkage, or a phosphodiester bond. Okay, so that's the structure and the function of all nucleic acids. And what we need to do now is talk about the difference between RNA and DNA. Oh, sorry, let me show you a picture really quickly of a nucleotide. Again, this is our sugar. So this is either going to have an OH, a hydroxyl group, or just a hydrogen atom. That will determine whether it's ribose or deoxyribose. If it just has the H, it's deoxyribose because it's been deoxygenated. If it has the OH, it has that oxygen, and so it's ribose. But this sugar, whatever it is attached to a phosphate and then some nitrogenous base. And you'll see that the nitrogenous bases exist in two forms, right? We have this 5-member ring attached to a 6-member ring. These are our purines, and they're either going to be A or they're going to be G. And then we have our pyrimidines down here that are just 6-member rings, and they exist as either T or C or U. So I guess just as an exercise, what we could do, right, is on an exam, I could give you some sequence of nucleotides, and then you could tell me what the complementary sequence was. So if I gave you the sequence A, G, C, T, T, A, C, G, G, K, and we know that A's always bind to T's and C's always bind to G's, then you guys could tell me what the complementary sequence was, right? So if A's always binds to T's, then the complementary base pair for this one is going to be T, right? Because A's always binds to T's. Here, G's, we know G's always binds to C's, and so the complementary sequence is going to be C, A, right? So that's pretty straightforward. So that's DNA complementary nucleotides. And I'm going to come back to this next point over and over throughout the semester. Lots of textbooks and lots of biology instructors say that complementary sequences are mirror images of each other, and that is just blatantly false. They aren't mirror images of each other, right? Because a mirror image looks something like this. It's a left hand and a right hand. If I was to put my left hand up in the mirror, what I would see is basically my right hand, right? It looks exactly like it, it's just flipped. My thumb is on the opposite side of my hand. But this isn't like that at all. These are complementary sequences. If you were to reverse that, you don't get the complementary sequence. You get a complementary sequence. And that's important, not just for the sake of terminology, but because really what complementary sequences do is allow us to double the amount of genetic information stored within the DNA molecule. Now think about that. If it truly was just a mirror image, and we know that DNA is only read in a single direction, so it's either read from left to right on the top or right to left on the bottom, we only have 50% of the information that we could otherwise have if the complementary sequence says something very, very different. So that probably meant very little to you. What I need you to understand, though, with this idea of complementary sequences is a complementary sequence increases the amount of genetic information stored within nucleic acids. And that's an incredibly important point.
Can you say that one more time?
You bet. Complementary sequences increase the amount of genetic information stored within the DNA molecule, as opposed to mirror images. Okay, so let's talk about the difference between DNA and RNA. So there is a primary structural difference between DNA and RNA, and that is the sugar, right? That's the first difference. So DNA has deoxyribose and RNA has ribose. Secondly, their shape is very, very different, and their function is very, very different. In RNA, there is no thymine. There's only uracil. So if we were to make an RNA complementary sequence to this DNA, instead of a T, we would write a U. So here, instead of A binding to a T, A would bind to U. The G's and the C's would be identical, so it would be C, G, and here the T would bind to an A, just like it does in DNA. But again here, we're replacing our thymine with the uracil, so that would be U, G, C, C. So this is an RNA complement, and this is a DNA complement. Unlike DNA, RNA is going to remain single-stranded. So we know that DNA looks something like this, right? So there is some alternating phosphate sugar backbone. For our DNA molecule. And coming off the middle kind of those sugars and phosphates, there are our nitrogen spaces, which are either A's, T's, G's, or C's, like that. But on the other side, there is a complementary strand that has the opposite orientation of its phosphates and its sugars. And it will have complementary nucleotide sequences, like so. RNA, however, is single-stranded. It will just be something like this. And that's how RNA remains. Now that doesn't mean that RNA doesn't form a three-dimensional shape, because it does. This long strand of RNA will fold in on itself, and those nitrogen spaces will hydrogen bond to each other to make really intricate shapes. An important example of that is something called tRNA. And tRNA is involved in the production of proteins. tRNA is actually what comes along and reads some copy of our DNA and attaches amino acids in the correct order. Additionally, there's an organelle within our cells called the ribosomes that we'll talk about a little bit later that has some three-dimensional structure, and it is primarily RNA. It's an organelle that consists almost entirely of RNA with some structural proteins. DNA, however, has two strands, and those strands spin or twist in space so that it looks like a ladder that's been twisted. Now, this is an aside, but it's an important aside. Not all hydrogen bonding between adjacent nucleotides is the same. Between A's and T's, we form two hydrogen bonds. But between G's and C's, there are three hydrogen bonds. Like that. And it's really just hydrogen bonding that's holding complementary strands of DNA together. So why is that difference between A's and T's and G's and C's important? It's important because the number of hydrogen bonds in our DNA molecule indicates stability. And there's some environments in which we want very high stability for our genetic material. There are some organisms that are called thermophilic organisms, archaea, single-celled bacterial-like organisms that live in thermal hot springs. So if you guys go, when you guys go to Yellowstone, you'll see these really rad hot springs, right? They have like these concentric rings of different colors, and they can be orange and green and blue and red. Those are all bacterial-like organisms that are living in extremely hot water. Now, those organisms have evolved to have way more C-G pairs than A-T pairs. And that's an adaptation because the more hydrogen bonding there is between adjacent nucleotides, the more stable that DNA molecule is going to be. And we know that heat can disrupt the stability of complementary strands.